Hello everybody. In our last video, we studied about the photosystems. In this video, we'll try to understand how the photosystems are used in the light reaction of photosynthesis. In plants, light reactions take place in the thylakoid membranes of the organelles called as chloroplast. As discussed earlier, photosystems, which are large complexes of proteins and pigments, play a key role in the light reactions. As studied earlier, there are two types of photosystems, photosystem 1 and photosystem 2. When the P680, that is a special pair of photosystem 2, absorbs energy, it enters an excited state. That means a high energy level. Excited P680 is a good electron donor and can transfer its excited electrons to the primary electron acceptor called as pheophyton. The electron will be passed on through the first leg of photosynthetic electron transport chain in a series of redox, or also known as electron transfer reactions. After the special pair gives up its electrons, it has a positive charge and needs a new electron. This electron is provided through splitting of water molecule, a process which is carried out by a portion of photosystem 2 called the manganese center. When the manganese center splits water molecules, it binds two at once, extracting four electrons, releasing four proton ions, and producing a molecule of oxygen. About 10% of oxygen is used by the mitochondria in the leaf to support phosphorylation, and you'll be surprised to know that the remainder escapes to the atmosphere where it is used by aerobic organisms like us to support respiration. When an electron leaves the photosystem 2, it is transferred first to a small organic molecule called plastoquinone, then to a cytochrome complex, and finally to a copper-containing protein called plastocyanin. As the electron moves through this electron chain, it goes from a higher to a lower energy level, releasing energy. Some of the energy is used to pump the protons from stroma into the thylakoid interior. The transfer of protons along with the release of H plus from splitting of water forms a proton gradient. ATP synthase harnesses the flow of protons to make ATP from ADP and inorganic phosphate. Once an electron has gone down the first leg of electron transport chain, it arrives at photosystem 1, where it joins chlorophyll A special pair called P700. Because the electrons have lost energy here prior to their arrival at photosystem 1, they must be re-energized through absorption of another proton. Excited P700 is a very good electron donor, and it sends its electrons down a short electron chain. In this series of reactions, the electron is first passed to a protein called ferredoxin, then transferred to an enzyme called NADP plus reductase. This enzyme transfers the electron to the electron carrier NADP plus to make NADPH. This is the diagram that summarizes the flow of electrons from photosystem 2 to photosystem 1. As you can clearly see in the figure, due to the absorption of the light, the photons get excited and reach a higher energy level, whereby they are transferred through a series of electron transport chain, including plastoquinone, cytochrome complex, plastocyanin, to photosystem 1, down to chlorophyll A, to ferredoxin, and then to NADP plus reductase. In this diagram, we can clearly see that two important molecules called NADPH and ATP are produced as a result of the light reactions. These two molecules are further used in the dark reaction or Calvin cycle of photosynthesis. Thank you so much for your patient hearing. In our next videos, we'll try to understand that how NADPH and ATP are used to synthesize and produce carbohydrates in the process of Calvin cycle. You can also visit my Facebook page or write to me on my mail ID. Thank you so much.